What if the secret to reversing aging wasn't hidden in fixing broken genes, but in rewiring the very instructions that tell our cells how to age? For decades, we have accepted cellular decline as an inevitable march toward decay. But emerging research suggests we might be looking at the wrong target entirely. Today, we are diving into a groundbreaking shift in longevity science that moves beyond managing symptoms to actively reversing the biological clock. Stay until the end because this new approach using safer gene editing tools could fundamentally change how we treat age-related diseases, though it comes with a critical caveat we must address. For generations, the field of gerontology has operated under the assumption that aging is a one-way street, driven by accumulated genetic errors and irreversible damage. We have spent decades developing treatments that manage the symptoms of aging, treating the wrinkles, the frailty, and the diseases like progeria that accelerate this decline. However, this traditional view often misses the deeper molecular root of the problem. The core issue isn't necessarily a permanent mutation in our DNA sequence, but rather a dysregulation of the epigenome, the complex layer of chemical markers that tells our genes how to function. When these epigenetic regulators go haywire, cells lose their identity and function, leading to the visible signs of aging. This distinction is vital because it changes the entire strategy for intervention. Instead of trying to repair a broken blueprint, we might actually be able to reset the software running on our cells. This is where the latest research offers a radical new perspective. Scientists are now utilizing a specific variant of CRISPR technology, known as catalytically inactive Cas9 or dCas9, to target these epigenetic regulators directly. Unlike the standard active Cas9 enzyme that cuts and edits DNA sequences, this inactive version acts more like a precise delivery vehicle, capable binding to specific locations in the genome without making permanent cuts. In recent studies using progeria mouse models, researchers applied this tool to modulate histone marks and chromatin states, effectively reprogramming aged cells back to a youthful state. Imagine it like a librarian who doesn't rewrite the books in the library, but simply organizes the shelves so the right stories can be read again. By targeting specific loci responsible for cellular senescence, this method demonstrated the potential to reverse age-related decline in living organisms without the risky off-target effects often associated with traditional gene editing. The beauty of this approach lies in its precision and safety. It locks in therapeutic changes by modifying the epigenetic landscape rather altering the underlying genetic code, offering a path to rejuvenation that minimizes unintended mutations. The implications of this discovery extend far beyond the laboratory, signaling a profound transformation for medicine, engineering, and our understanding of human health. For clinicians, this validates a future where therapies for conditions like progeria could move beyond palliative care to actual reversal of cellular aging, targeting the molecular roots of disease rather just soothing the symptoms. For bioengineers, it highlights a necessary pivot toward developing highly specific, low-toxicity protocols that prioritize safety and precision in complex biological systems. However, we must remain grounded in scientific rigor. While the theoretical promise of epigenetic reversibility is immense, current reports reveal a significant gap: the lack of concrete quantitative data. We do not yet have the long-term survival rates, specific methylation density metrics, or comprehensive safety profiles needed to fully validate the magnitude of this rejuvenation. This disconnect between theoretical potential and demonstrated empirical evidence serves as a crucial reminder that while the path forward is clear, the journey requires meticulous validation before widespread clinical application. Ultimately, this research represents a pivotal moment in our quest for longevity, bridging the gap between abstract genetic theory and practical therapeutic intervention. By leveraging safer CRISPR variants to modulate the epigenome, we are unlocking the possibility of reversing cellular decline and extending the human health span. Yet the story is not finished. The next chapter depends on gathering the rigorous data needed to confirm these findings. What do you think about the potential of epigenetic reprogramming to reverse aging? Is the promise of resetting our cellular clock enough to shift our medical paradigm? Or do we need more hard numbers before we celebrate? Share your thoughts in the comments below, And don't forget to like and subscribe for more deep dives into the science shaping our future.